This next session is all about one. I think everybody understands in this room. As you read the news, there are, big swings in pipelines. There's a lot of rare disease is a very volatile state. There's a lot of drug development going on. The investment in time, talent, and treasure for rare diseases, is an episodic journey of hopes and sometimes great sadness with drug development. And so one of the things I think when we talk about from a drug development perspective, obviously, one of the things that's near and dear to CPATH is the tools that we work in are all aimed at de-risking, drug development, rare diseases. And so, we wanted to have a session, for someone that has great experience in economics and financial modeling and give their perspective and really discuss things that and considerations around drug development and rare diseases. And we want to pass. And is Andrew on the line? I want to make sure. Okay. Do you want to go ahead and start his introduction? And okay. Thank you, Andrew. Do you want to go ahead and introduce yourself and you can start your presentation? We're all excited to hear, and we're thankful that you can join us, remotely. Thank you.
Oh, sure. Thanks so much, Colin. First of all, I want to start by thanking Colin Hovinga and the CPATH for inviting me to participate in this meeting today. And I want to apologize for not being able to be there in person and appreciate the opportunity to present remotely. I don't know if the AV folks can highlight or spotlight my presentation, because otherwise it'll be a little bit small since I'm not going to be sharing my screen in the conventional way. After a couple of years of teaching online, I've developed a different approach to doing online teaching using the so-called weatherman approach. This is the closest that we can come to simulate my being there right with you. So I hope this is okay. And for those of you who are watching online, you may want to maximize my screen just so that you'll be able to see the slides. So I want to start with a little bit of a disclaimer, which is that I am not an expert in biomedicine by any means. I got interested really from my main field, which is applying mathematical and statistical models in various investment strategies, hedge fund models, risk management, and so on. I got interested in healthcare really because of personal reasons. About 15 years ago, a number of friends and family started dealing with various kinds of cancer. And through that process, I realized that finance plays a really big role in drug development, sometimes too big a role. So what I want to tell you about today is some of the research that I've been doing since then on applying the tools of modern finance to drug development issues. And particularly in the rare disease space, that's going to turn out to matter a great deal. So let me start with an observation, which is that biomedicine is at an inflection point, and this audience knows this far better than I do. But the examples that I come up with are ones that I'm sure that you've come across as well, which is therapeutics that could not have existed even 10 years ago that today is becoming routine. And I'm going to give you two examples. One has to do with these two kids, Caroline and Cole Carper. They were born with a single gene mutation that causes blindness starting at birth, Leber's congenital amaurosis. And this is a picture that was taken shortly after their participation in a clinical trial that was in May of 2016 by Spark Therapeutics for a gene therapy that would fix this genetic typo. And a few days after the gene therapy, this is what Caroline had to say. I went outside when it was snowing and I was like, oh, I can see the snowflakes. It was really cool to actually see something that I've never seen in my life before. And of course, in 2017, Spark Therapeutics treatment for this particular disease, Luxturna, was approved by the FDA. The second example has to do with a company that's about 3 blocks from my MIT office, a company called Agilis Biotherapeutics. They were introduced to me because they had trouble raising funding for a gene therapy that they were working on, and I wrote a case study about them. The gene therapy is for a condition known as aromatic L-amino acid decarboxylase deficiency. It's a mouthful, and it took me an afternoon to figure out how to pronounce it. And it turns out that this is a single gene mutation that prevents infants from being able to produce dopamine in the brain. And without it, they're not able to develop normal motor functions. It turns out that a Taiwanese doctor by the name of Paul Hu developed a gene therapy to treat this particular disease. And what I'm going to show you is a video clip of patient number 4, who was diagnosed at 2 years and 5 months. And what you're going to see is the patient before the gene therapy, so the baseline. And then you'll see that same patient after this one-time injection of this viral vector into his brain, a year later, and then 2 years later. So, this is the baseline. And you'll see that the child is not able to raise his arms by himself. He can hardly breathe by himself, so he has a breathing tube. And he's not going to be able to roll over and do anything. This is what his life will be for the rest of his life. And now, the very same child, 1 year later, he's crawling. He's moving. He's almost standing, not quite, because remember, it's only been a year since he's started producing dopamine in the brain. And as he tries to balance, you'll see that he's not able to do that yet, but he's learning. And this is just a remarkable difference. Now, same child, 2 years later, after this one-time injection of the viral vector. And you can see that he's progressed. He's still not able to walk normally because it's only been 2 years now since being able to produce dopamine. But this is just an extraordinary achievement relative to what his baseline was. The blind shall see, the deaf the lame shall walk, and the blind shall see. This is the kind of typical, religious kind of experiences that are happening today, thanks to these incredible breakthroughs that are being pioneered by people in the audience and elsewhere. And so this is what I mean when I say that biomedicine is at an inflection point. My colleagues, Phil Sharp, Susan Hochfield, and Tyler Jacks, they wrote a monograph in 2016 describing this kind of inflection point, and they call it convergence, the convergence of the life sciences, physical sciences, and engineering. Other experts describe it as the so-called omics revolution. Every one of these omics has experienced tremendous advances over even just the last 5 years, never mind 10 or 20, with the exception of one. There is one omics that has been the bottleneck to progress in biomedicine. And what is that omics? That's economics, because you got to pay for all of this stuff. And we're using these same business models that we've been using for the last several decades. So that's what I want to talk with you about today, is how do we change the landscape of funding so as to address the issues of the Valley of Death. Now, I have to tell you that when I first heard about the Valley of Death, I was a little puzzled. I was wondering, where is this? Is Southern California? Well, why is this relevant for biotech? And then I realized what they were referring to is that a particular area between basic scientific research and getting drugs into patients in a phase 1 trial. That area of preclinical development is very, very difficult to fund. And so my question was, why? It shows you how naive I was. I just assumed, back when I first started looking into this, that if there were some patients in need and we had some biomedical technology that could help them, that magically money would appear to fund it and they would get their treatments. And I realized very quickly by looking at the situation with my friends and family that that was not the case. And after a number of years, I finally come to a conclusion as to why this is the case, why there is a Valley of Death. And it has to do with risk and uncertainty. Risk is the kind of randomness that you can predict to some degree, things like being able to calculate a normal distribution or mean and variance. And uncertainty is the randomness that you can't quantify, the unknown unknowns. And it's because of both of these features of biomedicine, the fact that they've been increasing in risk and uncertainty, not despite, but because of these scientific breakthroughs, that has made it more difficult to fund early stage drug discovery. And so I'll give you an example of what the underlying financial motivations are, and you can start to think like an investor, and when you do, you'll realize immediately what the problem is. So for my first year MBA students, I give them the following example. I show them 4 different financial assets. I don't tell them what they are or over what time period they span. I simply show them what happens if you invest $1 in each one of these and you leave it there for an unspecified multi-year investment horizon. The green asset turns $1 into $2, not very rewarding, but not particularly risky. The red asset turns $1 into $5, way more rewarding, but quite a bit more volatility. The blue line is the most rewarding of all, extremely volatile though, way, bigger ups and downs. And finally, the black line is somewhere in the middle. So by a show of hands, I'm going to ask you to pick one of these 4 investments for your retirement fund, for your kids' college education fund, for your parents' or grandparents' life savings. I want you to pick only one of these to manage your own personal financial assets. Which one would it be? I'm not going to see you, but the rest of the audience will. How many people, by a show of hands, would pick the green line? Any takers? Okay. How about the red line? How many people? Wow, that's interesting. So I'm going to have a few words about that. Not too many people raise their hands about the red line. When I tell you what it is, you're going to maybe want to call your broker. The blue line. Anybody for the blue line? Okay, those are the hedge fund wannabes and the biotech startup founders. And now, how about the black line? Raise your hand. Anybody want the black line? So my guess is that this audience, as every other audience that I presented this to, the vast majority will pick the black line. When asked why, they'll say, well, it's got the best trade-off between risk and reward. It's not the most rewarding, but it's certainly less risky than many of the other examples. Well, so let me tell you what you all picked. First of all, the investment horizon is from 1990 to 2008. And the green line is US Treasury bills, the safest asset in the world, at least for the next few weeks. We'll see what the budget discussions look like over the course of the coming few days. But assuming that we agree to pay our bills, US Treasury bills are very safe. And if you had invested in that in 2008, you would have earned pretty much nothing. The red line that most people do not pick, that's the US stock market, the S&P 500. I suspect that most of you already have that in your retirement funds. So if you didn't raise your hand, you may want to do some rebalancing and call your broker and cash out and put it to something else. The blue line, which very few people pick, that's the single pharmaceutical company, Pfizer. Oh, actually, before I get to that, if you picked the S&P 500, you would have done just fine. So, congratulations since 2008. But Pfizer, the single drug company that's way, way, way more volatile, if you had picked that in 2008, well, even bigger congratulations, you would have done spectacularly well, particularly through the pandemic because of the Pfizer-BioNTech combination. What about the black line, the most popular choice? Well, it turns out that that is the returns to a hedge fund known as Fairfield Century, which was the feeder fund for the Bernie Madoff Ponzi scheme, which is why I had to stop it in 2008. It blew up in that year. Like a moth to a flame, human nature is drawn to high-yielding, low-volatility assets. And in the case of the Madoff Ponzi scheme, now you know why it grew as big as it did. Because it just looked too good to be true. And the lesson is sometimes in life, things that are too good to be true really aren't true. But on paper, it certainly looked really attractive, as you all saw from this audience's reaction. In finance, we have a way to describe this phenomenon. It has to do with a metric called the Sharpe ratio, named after William Sharpe, the Nobel Prize-winning economist at Stanford University. The Sharpe ratio is simply the ratio of the expected return of an investment minus T-bills divided by the risk of the investment, in this case, the standard deviation of the return. So that measures the fluctuations, exactly what you all were responding to. We are all drawn to high Sharpe ratio assets. We want a lot more bang for our buck. In particular, we want a lot more excess returns per unit risk. That's what we're looking for. And if you look at the Sharpe ratios of the S&P 500 and Pfizer, they're about the same. 0.43 for Pfizer, 0.54 for the S&P 500. And on paper, before Madoff blew up, it had a Sharpe ratio an order of magnitude higher. Clearly unrealistic, but before we knew it, we were all drawn to this investment opportunity. So why am I showing this to you? It's because biomedicine has had a decreasing Sharpe ratio over the course of the last couple of decades. Not because the numerator has been challenged. There are a number of very successful biotech companies that have earned great returns for investors. The problem is that on average, the denominator ends up being even larger. It's even more risky because we don't know who the winners are going to be. And it's that risk and the fact that the advancing technologies, the breakthroughs that many of you in the audience are pioneering on behalf of the rest of us, those are wonderful things for patients, but an incredible breakthrough can mean that an existing drug that investors are earning reasonable rates of return on will be made obsolete overnight. So when Zolgensma was approved by the Novartis drug for spinal muscular atrophy, that was great for Novartis. It was great for SMA patients, not so great for Biogen and their investors that created Spinraza a couple of years before that. So that's the challenge. And if there's one thing that I can tell you about investors is that they will vote with their dollars. They will put money into higher Sharpe ratio assets and take them out of lower Sharpe ratio assets. And where the Sharpe ratio is lowest is in the early stages of drug discovery, namely that area of the valley of death. Now let me give you another investment opportunity and see whether you like this any better. Imagine an investment where you have to give me $200 million. Not you individually, but invest whatever you want, but I'm gonna raise a fund that's gonna have $2 million in it, and it's gonna take me 10 years to be able to generate any returns on that investment. And the probability that I generate any positive returns at all is 5%. 95% of the time, I will give you back nothing after 10 years, and that'll be the end. How many of you would you put your money in this by a show of hands? Well, I suspect that not too many of you. Now, when I show this to my MBA students, they have the same reaction that you do, not interested. But occasionally, I'll get one rather brave MBA student who will raise her hand and say, Professor Lowe, when you told us about this investment, you didn't tell us. In the unlikely event that there is a positive payoff, what is it? You see, most people don't need to know. When they see this, $200 million, 10-year waiting period, 95% failure rate, no thank you, not interested. It turns out that these are the back of the envelope numbers for what it takes to develop a single anti-cancer compound. It takes about 10 years to run the clinical trials. It takes about $200 million in out-of-pocket costs to pay for those trials. And the chances of success historically are actually a little bit less than 5%, 3.4% to be precise. I rounded up just to make the numbers easier. But in the unlikely event that you do succeed, it turns out that a single anti-cancer drug could generate, on average, about $2 billion a year in profits from years 11 to 20. Why 11 to 20? Well, because the patent life of a drug is about 20 years, and the first 10 years are taken up running clinical trials. So if you are lucky enough to get it approved in year 10, starting in year 11, you will generate that $2 billion from years 11 to 20, and when the patent expires, you fall off that patent cliff, and now your profits are very, very much lower. This $2 billion a year from years 11 to 20, that is equivalent in financial terms to a single payday in year 10, if you get approved, of $12.3 billion. So let me ask you again, anybody in the audience, are now interested in this deal, $200 million investment, wait 10 years, 95% of the time you lose everything. 5% of the time, I'll give you a check for $12.3 billion. Any takers? My guess is that maybe a couple more people, a few hands, are interested in it. But this is still way too risky. In particular, if you do the math, the expected return during those first 10 years of investment is going to be about 12%. But the risk, the thing that goes in the denominator of the Sharpe ratio, is 423.5% volatility. So this is a Sharpe ratio of 0.02. No, thank you. Not interested, right? So this is where finance can actually do something. Now, the failure that's going on here, if you ask what the failure is, it's really the fact that you're getting hit with a triple whammy of this kind of an investment. The horizon is too long, the costs are too high, and the probability of success is too low. And when you have all three of these things in one particular investment, I call that a long shot. Now, long shots can be very, very profitable. They can actually be very beneficial to society, but they are just very difficult to get investors interested in putting money up to invest in it. So what do? Well, the answer comes through a long, long list of biomedical research papers that I've published. And let me just tell you what we need to do. Two things. We need to increase the numerator of the Sharpe ratio, and or we need to decrease the denominator. That's it. It's as easy as that. Now I say easy. How do we do that? All of you, what you are doing, what CPath is doing, is exactly this. What you're trying to do is increase the numerator and decrease the denominator. What I want to show you now is a very simple financial engineering tool that can do the same thing. So instead of investing in one of these programs, what if we invested in, 150 of them, all at the same time? That's a lot of programs, granted, and it's going to cost us a lot of money. It's going to cost us $200 million times 150 programs, or $30 billion. Where are we going to get $30 billion? And as an economist, I have a very simple answer. The answer is, assume we have $30 billion. Now, you may be skeptical of that, but I'll come back to where I'm going to get it in a minute. But assume I have it. Assume I have $30 billion. And assume further that the programs are statistically uncorrelated. What I mean by that is that the success or failure of one program has nothing to do with the success or failure of a different program. Now, I know that's a tall order, especially in oncology, because if you've got 5 angiogenesis inhibitors and one of them fails and they're all targeting the same particular mechanism, if one of them fails, that's not good news for the other 4. But bear with me for a moment. If they are statistically uncorrelated, then when I look at the economics of this particular portfolio, look what happens. The standard deviation is going to decrease at the rate of the square root of the number of programs. So now my Sharpe ratio is 0.34. Remember, before this, it was 0.02. 0.02 to 0.34. So now, let me ask the audience, how many of you, by a show of hands, would invest in this portfolio? I suspect we have a lot more hands up. And that's where I'm going to get the 30 billion. Not from me. I don't have it. I'm going to get it from you. If I can change the economics of this particular investment, I can crowdsource this on steroids. I can get large amounts of capital to flow into these investments if I can change the economics. Now, can we really raise $30 billion? And the answer is, it depends. It depends exactly on the measurement of the various different risks and rewards of these portfolios. when I published this paper, one of the things that I was prepared for was skepticism. And it turns out that there is one area where this assumption of uncorrelatedness can actually be defended, because that's one of the weakest links in this argument. It's, this kind of uncorrelated nature allows you to really de-risk the portfolio, but is it likely to happen? It turns out that it is in rare diseases. You all know that there are over 7,000 different rare diseases, and many of them have nothing to do with many others because they're so different in terms of their causes, their mechanisms of action, the way that we interfere with these diseases. And so for a variety of reasons, it turns out that from a financial perspective, the economics actually works quite well for rare diseases, but largely for one very important reason that has nothing to do with science or medicine. And that is what I just described, which is the uncorrelatedness of the different diseases. Now, it's not always true. For example, in the cases of Leber's congenital amaurosis and AADC deficiency, they're very different. But if you use the same viral vector, in both those cases, it was AAV9 that was used, and it turns out that there's an immunogenic response, as there was when Bluebird was applying it for one of its diseases. The FDA will put a hold on all AAV9 therapies. And so there is correlation that you have to manage, but we know how to do that. There are so many different ways of treating these different diseases, whether it's through ASOs, gene therapy, gene editing, small molecule drugs, radiation, many, many different approaches. It is actually possible to construct an uncorrelated portfolio of rare diseases. So I wrote a paper about this in 2015, published it with some collaborators from the National Institutes of Health, the NCATS in particular, the National Center for Advancing Translational Sciences. They have a rare disease unit, and we simulated a portfolio of rare disease therapeutics and found that that portfolio would not only be successful at developing a number of drugs, but it would generate a rate of return on the order of 22% per year for the life of that particular fund. Now, this is a theoretical finding, and so undoubtedly you might be skeptical. And there were a number of people that were. But one of my former students from MIT contacted me. He read the paper, and he said, first of all, I really like your analysis of rare disease therapeutics with these portfolio kind of diversification arguments, but would you mind rerunning your analysis with my assumptions instead of yours? Because obviously the parameters that I had used for running the simulations were from the industry publications. I had no particular expertise in the area. The work that I did with NCATS, they used their data and then industry figures. We used his figures instead, and over the course of 6 months, we must have run 35 additional simulations. Each time, the results kept getting better and better and better. And one day, he came into my office about 6 months after we started this exercise, and he said, I just wanted to let you know, I quit my job yesterday. I'm gonna do this. I'm gonna build this company. And I gotta tell you, that freaked the heck out of me. I'd never had that effect on any student before. And he had a young child in his household, so I said, Neil, you sure you want to do this? Maybe you better let me run a few more simulations. And he said, no, I'm really excited about it, ready to do it. I felt guilty enough that I invested a small amount of money in the friends and family round. And in retrospect, I wish I had invested a lot more, because the company called Bridge Biopharma has its business model focused on rare diseases. And shortly after we came out of stealth mode in 2017, we raised $40 million from investors because of that portfolio structure. A year later, $135 million. A year later, $300 million. And in 2019, we did an IPO, the largest biotech IPO at that time, and also raised some debt financing. And as of a couple of days ago, the market cap of BridgeBio is a little bit more than $4.5 billion. Not bad for a 7-year-old company. But this is not what BridgeBio is most proud of. What the people and I are most proud of is this. It's the fact that BridgeBio now has a pipeline of more than a dozen assets in a variety of different rare diseases. And in 2021, it received its first FDA approval for a drug for cholangiocarcinoma and another one for a particular kind of enzyme deficiency, and breakthrough designation for other diseases, and just recently, a very big phase 3 readout for a disease that affects the heart, transthyretin cardiomyopathy. Without the portfolio approach, it would have been difficult to convince investors to be able to fund it. But with the right structure, with the right kind of financing, it is possible. Now, there are other business models that have emerged, including venture philanthropy. And in a paper that I published in 2022 on using different models like the Cystic Fibrosis Foundation, the Leukemia Lymphoma Society, multiple myeloma, and so on and so forth, there are ways for public and private organizations to work hand in hand. And a really wonderful example of that is what you are all doing at CPATH. This is a great example of increasing the Sharpe ratio by basically convening various different stakeholders and generating data. So I want to give great kudos to all of you at CPATH for doing what you're doing, and would love to support it in any way that we can at MIT. So let me wrap up, leave a little bit of time for Q&A if there is some, by pointing out that I started down this path for personal reasons. And over time, it's become even more personal as I've seen just how important these kinds of financing models are for helping patients. This was brought home to me in a very, very concrete way by one of my colleagues, Harvey Lodish, at MIT. Harvey is a cell biologist at the MIT Whitehead Institute. And when I first met Harvey and heard about his story, I decided right then and there, I want to be Harvey Lodish. And when I tell you his story, you're going to want to be Harvey Lodish too. In 1983, Harvey was an assistant professor here at MIT and was approached by some of his colleagues to help them work on a disease known as Gaucher's syndrome. So Gaucher's is also a single gene mutation. It prevents your body from producing important housekeeping enzymes, and without it, fatty acids build up in your organs, in your bone marrow, and by the time you're a teenager, if you have a particularly serious variant of this disease, you're dead. So this is not a small problem for the people that are afflicted with it. And in 1983, Harvey and his colleagues started out to develop what ultimately became the very first enzyme replacement therapy, which was Ceridase, approved in 1991. And that drug and all of its various improvements have saved literally hundreds of thousands of lives for patients that have Gaucher. And you might have heard of Harvey's little startup. It was called Genzyme. And in 2014, it was acquired by Sanofi for about $20 billion. Now, that's not why I want to be Harvey, although that's not a bad reason, particularly as an economist. I applaud that. The reason I want to be Harvey Lodish is what happened in 2002. You see, in that year, Harvey's daughter was pregnant with her second child, Harvey and his wife's second grandchild, a boy named Andrew. Great name, by the way. Andrew was diagnosed with Gaucher's in utero. What are the chances of that? And so I asked Harvey, it was a very emotional conversation. I asked him, Harvey, did you know in 1983 that you would be helping to develop the drug that would save your as yet unborn grandchild? And Harvey said, no, I had no idea. In 1983, it was 20 years before we even sequenced the human genome. I didn't know that I was carrying the gene for Gaucher's. I just thought if I could do some cool science and help some patients, that would be great. And so in 2012, when Andrew turned 10, he developed the full-blown symptoms of Gaucher, but he's leading a totally normal, healthy life thanks to the drug that Grandpa helped to develop. This is why I want to be Harvey Lodish. I will never be able to develop a drug that will save my as yet unborn grandchildren. But I realized something. I realized that all of us, we can all be Harvey Lodish if we invest in the drugs that will save our as yet unborn grandchildren. Finance doesn't always have to be a zero-sum game if we don't let it. With the right kind of business model, the right kind of science and medicine, the right kind of financing at the right scale, we can all do well by doing good. And with your help at CPAth, we can do it now. Thank you.
I guess, do we have any questions from the audience for Andrew? Anyone? Regular? Investment fund strategies? Okay, go to you.
Well, you just have to tell us what you invest in right now.
Sure, I'd be happy to. Two things. One, my retirement funds are, all of them, invested in low-cost, low-fee mutual funds at Vanguard, and usually broad, diversified portfolios like the S&P 500 or the international version of the S&P 500. My other investments are all in the area of healthcare. I'm investing in biotech companies, like BridgeBio, but others. And that's very risky. But my retirement, not at risk. I know I'll be able to retire and be able to, buy ice cream cones for my grandkids. But the area, the money that I can afford to lose, I'm investing them now in biotech companies that are really risky, but have the opportunity to transform people's lives. And if they do, they'll make a lot of money for all investors.
So I want to say thank you for making drug development sound fun and funny. That's not how we live, but okay, sure. But one thing I do want to say is, I mean, I know Harvey, and I know that story, and that was the best story until Bob Langer, right? Moderna.
Yes.
Like, he cried at the idea of the vaccine, but I mean, I really appreciate the story, and I wish you would teach that to all your other friends and family at academia, because we still have that battle trying to appreciate the value of what we're doing.
I know, and if I were there in person, I would have spent a little bit more time on the introduction part. It's a little hard when I can't see the audience, but I start almost all of my presentations first by thanking the audience for doing what you do, because it's not something that I can do, and you guys are all saving lives, and it's really important for the rest of us. So I'm very grateful for that, and a day doesn't go by when I don't realize just how amazing this profession is. That's one of the reasons why most of my research is now devoted to healthcare finance, because this is sort of the least I can do to help all of you do what you can do.
Do you have a question?
Yes, this was a really interesting intellectual exercise. Thank you for that. And going back to the economics, I was thinking, obviously, when you gave the first example that you invest $200 million on a risk asset, and then your potential reward is $2 billion a year, right? And then obviously you multiply it by 150 investments to reduce the risk. But when you go into the rare disease space, we're talking about small populations. So
right.
To attain your potential target, likely drug prices will be over the roof. So how do you expand on the economics of that?
Well, actually, so there are a couple of different factors that are important to keep in mind for rare diseases. One is that it doesn't require $30 billion. So I didn't have a chance to get into the details, but precisely because you have smaller patient populations, what that means is that running a clinical trial isn't 200 million, it's more like 10 or 15 million. So the economics actually changes all the way around. Now, you do bring up an important point, which is prices. It is true that rare diseases, in general, have higher price tags than more common diseases. But the reason is actually pretty straightforward. It's because it costs a certain amount to do the research. And whether or not you're developing a drug for a rare disease or a common disease, apart from things like infectious diseases where you need 30,000 patients for a phase 3 trial, apart from those examples, the cost is not that different for developing a drug for a rare or common disease, except for the numbers of patients in the clinical trial. So what that means is that when you have small patient populations, it costs less to develop the drug in terms of the clinical trial, but you're also going to earn less because you've got fewer patients. So what do you need to do? You need to increase the price per patient. And that's what Genzyme figured out. Prior to their launch of an enzyme replacement therapy, nobody would dare charge $200,000 or $300,000 per patient per year. But they said, look, if you want to save these lives, this is what it costs to develop the drug. This is what we need in order to earn a reasonable rate of return. And meanwhile, we are actually saving lives. This is not toenail fungus treatments. We're talking about life or death. And so from an economic point of view, it actually is a reasonable trade-off to be able to pay those prices to save those lives. So I would say that actually the prices of these gene therapies and other rare disease therapeutics, they're actually not nearly as problematic for payers and for the government, for politicians. They're not nearly as problematic as the price of insulin, which is really outrageous. Insulin's been around for a long time, and there are people that this is not a rare disease. A lot of people get affected by that.
Do you have a question?
Hi, Andrew. Bruce Bloom here. Thanks for a great presentation. Can you tell us what's the impact of AI going to be on the kind of work you're doing, especially like on the Sharp ratio and other kinds of things?
Yeah, that's a great question. In fact, AI is changing the Sharp ratio as we speak, not necessarily through the numerator, but through the denominator. So as I suspect this audience knows far better than I do, there are a number of AI companies out there that are engaged in drug discovery, and what they're able to do is to be able to identify targets that previously we were not able to identify as easily. And in some cases, they can predict toxicity in ways that existing methods don't predict. There are a couple of case studies that I know of situations where you can use these kinds of AI tools to predict toxicity in humans, where animal studies have shown that there isn't any toxic effects. So I think it's really remarkable what we're seeing right now with the power of AI, and certainly with programs like AlphaFold that are being able to generate three-dimensional structures for proteins on demand. I think that we're in this inflection point for at least another 5 or 10 years, where we're going to see some tremendous progress and therefore benefits from the financial side for investors in that time frame.
Last question.
Going back quickly to the question about payers, particularly in long-lasting therapies like gene therapies, if you mentioned that payers may have less of a problem with the cost, but when in our healthcare system, you're switching insurance companies and a company is investing in you $3 million for a treatment that they may not see the return, how do you see that playing within the economics of rare disease drug development?
So that's a fantastic point to end on, because there is a problem, a really serious problem, and I believe I have the solution to that problem. So what this questioner was referring to is the fact that a gene therapy like Hemgenix, that's the gene therapy for hemophilia A, that's currently the most expensive drug on the market and the most expensive drug in the history of the biopharma industry. $3.5 million to cure you of hemophilia. Now, it turns out that that's actually a bargain in the sense that hemophilia patients need to be treated now anyway. And one episode of a bleed and infection can actually cost, in terms of hospitalization and other treatments, it can actually cost about $900,000 for that one episode. So hemophilia patients are actually going to do better by the healthcare system if we were able to cure them. But the problem is that upfront payment can hit employers and insurance companies really, really hard. So there's a solution. The solution is, instead of charging a single upfront price, what we ought to do is to create a Netflix model, the kind that was used by Louisiana for Sovaldi and Harvoni, where you basically charge everybody in the healthcare system a small amount every month for the privilege of receiving Hemgenix at no additional cost should you need it. That subscription model is now currently being developed by a startup that I co-founded a couple of years ago called Quantile Health. And right now, it's not as big of a deal because the existing gene therapies are for relatively rare indications. But we have a gene therapy. We have 2 gene therapies in the works for sickle cell by Bluebird and by Vertex. And so when those get approved, and looks like they will get approved, this is not going to be a small problem. So the subscription model will provide a way to make it affordable and to make it broadly accessible.
Okay, I'll close the session. Thank you, Andrew. I hope family is all doing well, and I look forward to speaking again shortly. Take care.
Thank you, and thank you all at CPAC for doing what you're doing.
It's going to be a little bit hard to follow that one, so apologies. Next we're going to move on to a showcase for RDCA-DAP. So looking at data empowering research and machine learning. So would like to welcome up Kara and Laura from Iridia, which is the platform that we utilize for RDCA-DAP, and Ian Braun with CPath.
All right, we might need to do a little rejigging here, so give us a second, because we have some exciting stuff today. So we have a laptop. All right. So hello, everyone. Thank you very much. Thank you for the mic as well. So we're going to be talking to you today about RDCA-DAP, a platform you've probably heard mentioned at the conference so far. Maybe you've seen it, maybe you've used it, maybe you have no idea what it is. Just curious by show of hands if anyone has actually seen the platform or is a user, is logged in before, that kind of thing. So it's not many, if you look around the room. So this is quite cool for us because Laura and I, we'll talk about this in a moment, kind of what we do and where we're from. But we're going to be talking about the platform, and we also have Ian here from CPath, and he has firsthand experience as well, especially from the data side. We're going to be talking about fair data, why that's really powerful, and machine learning is here. It's a buzzword these days, but we're going to talk about how this data is not just sitting by itself on a platform, but by combining data and people and tools, it can become a lot more powerful together, and that's really the whole purpose of this platform. So it's also a platform that's not just limited to researchers and data scientists and pharma and biotech. This is something that is also for the community itself. It's meant to have patient input. It's meant to have interaction with the wider rare disease community. So we also want to encourage everyone to just, feel free to take a look at the platform because it's open for a reason, and it's FDA funded and it's shared and it's advocated here by CPath for a reason. We want to have this dialogue. So without further ado on the intro here, let's see if it's playing ball for us. There we go. So what we're going to be talking about today, I think we might need to make sure we're sharing things properly. There we go. All right, cool. So we'll let that get sorted there. But what we're going to be talking about today for RDCA-DAP is built on a platform by Iridia. So Iridia develops platforms for digital research in a cloud environment. So it's something that globally people can connect with. They can collaborate and do work safely and securely. And most importantly, they can use that to drive better patient outcomes. So for myself, I'm at Iridia. I'm Kara Lassiter. I'm the head of customer success. And for RDCA-DAP, I'm kind of the technical lead of all things platform and technical side, but also trying to help align things with that wider rare disease community. So very plugged into that. Laura?
Hi, my name is Laura Shishodia. At Iridia, my job title is product manager. I know in pharmaceutical, in medical sphere, that can mean many different things. At Iridia, it means my job is to gather feedback from all of our users across both the RDCA-DAP and our other installations of the DRE, and make sure the feedback from the end users, from the stakeholders, is being fed back into the product and that it's being released into the world for use, so that our product is doing what our users need when they need to do it.
I'm Ian Braun, so I'm a data scientist working at CPath, and I work on the curation workflows associated with getting data from contributors, curating it, applying automated workflows where necessary, and getting it onto the platform.
All right, and I'll turn it over to Laura to talk a little bit more about what the platform really is.
Oh, I can use that. So another show of hands. Who before this conference had not heard of the term FAIR in the context of data sharing? A few. Oh, more than a few, okay. So FAIR data sharing is, it's not just an Iridia or a CPath thing, it's much wider than that, and it describes a means of gathering and sharing your data that makes your data both findable, accessible, interoperable, and reusable. When we talk about findable, we mean that other users could maybe come and they could find and search for your data. Accessibility in the context of data means that you can request that data if you want to use it, and that you can then have access to that data to use it as you need it. The meaning of interoperable essentially comes down to your data being well described. Could a fellow researcher come along, see your data, and understand what it's for and how they could use it? We've heard a lot of instances over the past few days of the wrong data being available and you not having access to quite the right data. Maybe if the data was well described, you know exactly what you're looking for. The reusability kind of speaks for itself. In our context, it means that when you're using the data in a project sense, when that data is maybe returned to the pool and maybe used in a further context down the road, that someone else could come along, they could see the results of your analysis, they could maybe rerun it, that kind of thing. So that makes up one side of the DRE, and it's having that data in a safe, stored manner. And the other part of the DRE, so the other part of the RDCA-DAP platform, is what we call the workspaces. Workspaces are essentially a virtual laboratory for your team or your project to work in. They are secure, so the data is served into this virtual environment, and only your trusted colleagues are allowed access to that data. It comes with analytical tools, which we will show you shortly. Oh, we didn't mention we're going to attempt a live demo today, which is keeping us all on our toes. So we'll show you some of those live analytics, and what we won't quite show you today is the scalability as well. So you don't have to invest as a project in lots of long-term hardware, maybe kind of high-level compute. We can provide that on a kind of rental basis. And the last thing that our workspaces allow is accessibility from across the globe. So you can collaborate with colleagues, with maybe with people who are reviewing your science, for example, in any country. And this also means and supports data sovereignty. So you as a researcher can access data that you maybe wouldn't be allowed to because of where you physically are. So both of these parts of the RDCA-DAP platform we will show you later today. Cara.
All right, and then I think you've all seen this before, but it's kind of worth putting this in the context of what the platform is and what it does. So we kind of envision RDCA-DAP as having this kind of flow of data, especially like Ian was mentioning on the curation side. There's a lot of work that's done to curate data to make sure it's kind of standardized. It's something that is also enriched over time. There'll actually be a presentation, I think, right after this, so a little plug for that on another way that you can enrich data with ontologies. And it makes it possible to try to connect data in different ways, novel ways that haven't been attempted or been possible to do before. And that's only possible if you have this kind of robust framework for being able to work on the data and share it and be able to publish the iterations over time. So the platform facilitates that kind of framework for being able to publish that and share that and also be able to control the access, which is quite important. Because the data is something that is important to share and to remove some of those silos and barriers to access. But equally, it's important to make sure it's secure and it's audited and all of that's accounted for and the provenance is tracked throughout the whole way. One of the other ways that this is kind of an important part of the platform, like I said, from that patient and patient advocacy side as well, is greater levels of transparency into what data is out there, what's being shared, what the impact is of that data, how it's being used. So there's a lot of tools built into the platform, especially on that FAIR data side, that tries to track and capture that so there's a better level of understanding and clarity there. And it also helps you visualize and kind of get a sense of the stats and data sets that are available without needing to have access to the field level data itself. So it just brings a lot more transparency to that process across the board. And then, of course, the outcome of all of this is trying to find, advanced models for drug development, disease progression, better understanding of that can also lead to new solutions, new therapies, new drugs, and actionable, insights that actually feed back into the community. So that's kind of underpinning the whole idea. But as we've said, this all exists. So it's really cool to see it, and that's one of the reasons we want to dive into that a little bit more today. And before we go into that bit, it's worth kind of stepping back and just thinking about what's the purpose of this. And there's a couple of central goals here. So I think I'll ask, especially for that data perspective, for Ian to kind of describe why this is a crucial gap to fill.
Sure, yeah. So one of the central goals of the initiative is to create this platform for rare disease data sets that can sort of act as a storefront for publishing and managing this data on a global scale. And then also, critically, the platform is also designed in a way that the data and the metadata that describes it can adhere to these four FAIR principles: findability, accessibility, interoperability, and reusability that we were mentioning earlier. So the FAIR platform itself is critical to making data findable and accessible by providing sort of a central location where users or researchers can come and search for data, go through the metadata that describes the data, and use some of the tools that we're going to talk about in a bit to figure out if that data indeed fits their need or their use case, and also manages accessibility by providing these workflows for requesting access and having the data delivered to a secure workspace. The curation that goes into adding data to the platform from the CPATH side, once it's contributed to CPATH, contributes to the interoperability, because we go through a curation process of making sure that the tables and columns and field level values of the data set are described in a way that makes it easy to apply whatever techniques or analyses you need to bring to the data set for your particular use case.
And as I mentioned before, one of the key aspects of that is that kind of that audit trail and the security along the whole route of that. So one of the other goals of the platform is to try to make that as easy as possible, especially for data owners and data stewards to be able to manage who's accessing the data and what's being done with it. And by trying to alleviate some of that burden, because it can be quite a high administrative burden to managing a lot of data, so trying to automate some of that and try to make data access something that's built into the platform for identity and information about projects and usage for that data, and making it easier to manage that kind of record of what data sets have been accessed for what projects over time, and being able to say that all of that's been governed by those rules of terms of use and access conditions, and all that can be attributed back to the data that was used for whatever models or results were generated.
And if we look at the fourth point here, one of the things that we've heard time and time again over the last day and a half or so is that it really does take a collaborative effort to make strides in any research area. What we hope to provide as a central goal in the platform is that area where you can really collaborate with your colleagues and with your other researchers. The platform allows the data to be stored in one central place, and it allows you to control access to that data to the right people. But it means those people don't have to be sitting next to you. They don't have to be even in the same room as you or the same continent. They can access your data, the data that they have access to, from wherever they are, whenever they need. It also allows you to bring experts into your project at the right time. So, for example, if you've got initial access to the data, but data science isn't your specialist, you can bring your data science colleague in to give you a hand there. Maybe further down the line, you're wanting to take your results to the FDA, you can actually bring them in as well. And they can see that data in its space, run the same analysis, and show it live to the people who need to approve that drug or that research at the end of its life. And finally, supporting the key goal of any project, which is publication and sharing your findings and your results, whether they be good or they be bad. The state of your workspace and the state of the DRE is maintained after your project. So should you need to come back to that in 3, 4, or 5 years' time, when you've got your next iteration of that drug development, you've got the next iteration of your project, you can come back and find everything exactly as you left it. As Kara mentioned, the access to the data is audited, and so you as a data owner know who's got access to your data and where it's been used. But the actions that you do on your data are also audited, and they're also controlled. And this means, again, you can look back at what you've done, and you can kind of refresh your memory, and you can build on those results and keep making new strides.
All right, and I'll just briefly mention this because I think we've heard a couple of timelines and a couple of kind of history lessons today on various initiatives for rare diseases. But it's worth kind of pointing out that the kind of journey for RDCA-DAP started years ago in 2019. And it's been a collaborative effort between CPATH and FDA, as well as between especially the groups that have worked with NORD and CPATH and have contributed data and have kind of fed into this initialization of the platform. And in terms of the data itself, I think Ian was mentioning, there's a lot that goes into the curation side. So I'll kind of put a bookmark on that because I want to make sure we have some time here for demonstrations. But one of the things that also came up is in 2021. That's when the platform was first launched. So we've been getting feedback from 2021 with kind of early beta testers. And then later that year, it was publicly launched. So it's been available since then. There's been a lot of engagement from researchers and industry members and academia getting involved and getting involved with the data and research initiatives on the platform itself. But I also want to kind of call out here for Ian, if he wants to mention a little bit about CPONT, that's mentioned here.
Sure, and we'll hear more about CPONT in the next talk, but basically this is an ontology that we've built from existing vocabularies to help standardize our data sets internally and increase their interoperability. And sort of importantly, this is also what's the basis of our current efforts to expand on how researchers and users will be able to explore the data on the platform or query about what sort of information is contained within the data sets on the platform. So for example, something like coming to the platform and being able to find all the different data sets that contain a patient with a particular phenotype or a particular phenotype that is caused by a particular gene, et cetera.
And one of the things too that's been really good to see as the platform has been launched and there's been more engagement with the community here, there have been more and more data sets that have been added to this data catalog. In 2022, 64 data sets and over 24 diseases. And that kind of brings us to today, where we still have more data, more diseases being added, and we also have research projects with their own dedicated spaces and they're kind of like your own lab environment where you've got your tools and your data and your team in it. So there's a lot of kind of changes over time where research projects are spun up and spun down. But one of the important things here is that this is providing that framework for all of that to kind of come and play under the same environment here. And what happens next, that really depends a lot on the engagement that we keep getting from the community, because the platform is ever changing, it's ever developing and improving. We get a lot of feedback, especially from the CPATH team, about data curation and how to make data more available. But we also get a lot of feedback from the users, the researchers, and the patient advocacy side about how this platform can be more useful to them. And so those kind of things are constantly featured here. And we'll actually end today just showing a snippet of some of the things to come. But the most important thing here is live demo. So we've got things pulled up. And what we're going to show you today is kind of two different perspectives here at play. One of them is the patient side, where you're looking at, like I was mentioning earlier, what's on the platform, what data is here. Maybe you're interested in contributing or getting involved somehow. So I'm going to tell you a little bit about that and show you what that kind of perspective looks like. And then Laura's also going to talk about the research side and what it looks like if you're not only using data on the platform, but diving into some of those analytical tools. There's a lot built in, but it's also a very kind of extensible platform, so there's a lot involved if you want to get more advanced. So that's where the machine learning comes in, and I'll show you a little bit today about how machine learning on the platform, can be done, and it's I say simple, and that's not to trivialize it, but it's something it's quite cool to see because it does kind of show the power of this platform, especially if you bring all the people and the data and the tools into the same environment and give them the freedom to be able to do what they do best. So I'm going to flip into a browser here, and let's see if we can get things pulled up. All right, so like I said, we'll talk about the patient side first. So what I'm showing right now is the RDCA-DAP portal. So this is both kind of an information portal. It's got lots of good info here about the whole purpose of the initiative. We're all, I think, familiar with that here today. But one of the things that's also useful here is it gives you some guidance about what the platform is, how to use it, what your kind of first steps are if you're interested in getting involved. If you're looking at this as well, particularly from kind of the patient side, like I mentioned a moment ago, you might be particularly interested with how you can contribute data. If you're someone who has, or an organization that has research data sets, studies, clinical trials, and so forth, you can reach out to CPath and you can connect with them and let them know what you have and that you'd like to be involved. One of the other things here too is just some guidance that if you're a single patient or a patient advocacy, kind of persona here, there's other resources. You can get involved with registries like NORD, and you can help, contribute and feed this growing body of knowledge. So if I was interested in that, there's, kind of a simple form here, and you can just, get in touch really instantly with CPath, and you can provide some info so they can better understand what kind of data that you have and how you want to contribute that. And then if you're interested in what data already exists, we can take a look at Fair Data Services here. So this is, as Ian mentioned, this is kind of like the shop front. We've got all the data sets, all the metadata. It's all here to be browsable, searchable, and findable for you. So this is kind of the front landing page that you'll see here on FAIR. It very helpfully as well talks about the FAIR principles. So if you'd like to do some evening reading, you have all about that there. And then if you're interested in the data sets that are at play, you can browse all the metadata of these data sets and learn a little bit more about what's here. So we've got names and description. I can also filter this if I wanted to look for some of the most recent data. I can edit that. And I can also set it so I'm looking at data that's made available on the platform for people to view. So if I see a data set here, so this is Mimic. This is, I think, a pretty well-known EHR data set. So what you'll see kind of right off the bat for any of these data sets is just some descriptive information about what's there. Descriptions, authorship, publishing information, things about the license, if this is something that is open access or if you need to request approval for it. There's some good information there that just kind of describes what you're looking at for all these data sets. And then when it comes to the data itself on kind of a field level basis, you can see what information is describing the actual fields here. So there's usually multiple tables that make up a data set. And if you wanted to see what was inside those tables, you can start to, kind of browse through that and you can see what these fields are. And you can see what types they are, and if they're potentially something that is requestable, or if this is something that you can preview and do some basic kind of stats and visuals on. You can also come back to searching for like really specific things. So if I was to search for a data set rather than just kind of browse the whole catalog, there's a bunch of filtering kind of options here built in. So if I wanted to look for something specifically that was already set for our cohort builder visualizer, I can select that option. And I've got a couple keywords here about different things that have been tagged on data sets. So I think I'll go through some demographics here and we'll show an example with cohort builder where you can start to visualize some of that data. So we've got a synthetic clinical trial data set here. About PKD. And similar, thing, we've got a lot of information here. I can go straight into the cohort builder. And like I mentioned, the cohort builder is something that is great for visualizing and getting a preview of data. I can also build in really specific filters and create cohorts of data and subcohorts, or I can just see what kind of data is at play. So if I wanted to see what kind of a breakdown was of sex for this particular data set, it starts showing me visuals right off the bat. And I've not had to access the raw data in order to understand what's present here for this data set. So I have a cohort that I've already saved to help save on some time. And for this cohort, I've already got it kind of broken out for kind of a female subset here. And if I wanted to go to kind of a breakdown of what the spread was for this particular clinical trial, I can look at some of the demographics and the breakdown here. Let's look at race and let's look at age. So this is kind of the the real-time breakdown of this data, and I can also customize some of these cohorts. So if I wanted to create a second cohort here with kind of a male selection on that. So now I can see, just at a glance there pretty quickly, I can see what that kind of demographic spread looks like for this data set, and I can maybe even see that there's underrepresented populations here, or if the spread looks to be really good. And I can save that, and I can share that with friends, colleagues, family that joined me on the platform here. So I'm going to send this to Laura, and I'll let her view that cohort. So that's how that can be kind of used, especially like we're mentioning from that patient perspective of interacting with this and interacting in that dialogue with CPATH. But what happens if you're a researcher? I'm going to turn it over to Laura to show a little bit more on the analytics side.
Thanks, Cara. So like Cara said, we're now moving from the persona of the patient to the persona of the researcher. So this is someone who is coming along, maybe they've had a colleague flag up some data to them and they've asked me to look into more information about this data. So again, I'm landing on the same page. This is a portal which is for all users. It's not tailored to a particular persona. So it's giving me the services I need as well. As a researcher, I know the first thing I need to do is create that workspace that I can bring my data into. This can be easily done using this link right here. It is a very quick 7-question form, which once you've filled out and you have submitted, that will actually get sent directly to the CPATH team. They can then get back in touch with you if they've got further questions and they want to discuss your project a bit further, but otherwise, they will provision you with a workspace very quickly. Usually, I think it's 2 to 3 days, if I'm correct, around that. So you can access this data relatively quickly if you need it. So assuming that that's happened for the purposes of today, I now think that I want to go and find that data that's been flagged to me so that I can see those cohorts and I can actually move that data into my research workspace to get a finer view of it. So I'm going to jump into FAIR again. And I don't know if you can see, but in my browser here at the top, I'm working now in this orange space. And it just means that that's actually slightly segregated from the one Cara was looking at, just so that you know it is slightly different. We're not looking at exactly the same thing here. So I know that my colleague Cara has sent me this data set. So I should actually be able to see it if I look in my cohorts. And I think it was that top one, wasn't it? Or was this? Oh, there we go. So again, I can go in here and I can see exactly what she sent me. So that's really cool. And it's a nice way of collaborating, like Cara said, before you have real access to the data. So I can see that view. I can actually kind of build on this as well, and I can get maybe a different view that I need to. But in this case, I'm going to jump to my searches. So again, Cara could have shared her search with me to send me the exact data set that she wants to collaborate on. Again, I'm jumping into the same thing, and as Cara showed earlier, there's like a lot of good information here that is available to educate me about what is in this data set. So knowing that I want access to it, what I need to now do is make a request. This is done very easily, again, using a form which is within the platform. I'm not going to go through this step by step because it's a slightly longer form than last time and it's not particularly interesting to show you. But again, it's just asking for information about your study, about yourself, about your credentials, just so that we know, or the data owner knows, that the data that they're sharing with you is going to the right people. We have a 4 safes model, and one of those safes is that we have trusted people who are accessing our data. So we just need to check that the people accessing the data can be trusted. Once you've popped your information in here, because it's things like your contact details, again, if the data owner or the CPath curators need to get in touch with you, they can do that. Once I've submitted this form, there's a background process which goes on where the owner of the data or the assigned person at CPath will then actually give you access to that data on a per data set basis. So I might actually go in and request 2 or 3 different data sets, but if that owner doesn't want to share that data with me, they don't have to. So as maybe a trial participant, as a patient who's been part of a previous study, you know that your data isn't just going to anyone, it's only going to people who those, the people who run the trial really trust with that data. So, having accessed the data, and again, let's assume for the purposes of today that I have been granted access, I can see a list of all the requests for data that I've made. And the one that we're looking at is this top one, which we've prepared. So this is, what I'm doing here is I'm now ready to transfer that data into that workspace that I requested earlier. So if I start that now, and the data gods are kind, we should be able to see the data being delivered into my workspace. So we've now gone from FAIR services, if you remember I talked about earlier, into the workspace sphere of the DRE. Each workspace is kind of like a project in itself. So this is your project area. You can see in this example, I'm maybe a member of 3 different projects. But for today, we're going to look at my research demo project. Oh, did I click on it? I did not click. There we go. So when I land in here, my project has been set up so that it's got some information, it's got some useful links here. I can see a list of my collaborators, so I know that I'm collaborating with the right people. If I have the right credentials and the right level of authority, I can also invite other people into my project as well. My data, which is delivered, is going to end up in my inbox, unsurprisingly, so I can click in here, and I can actually see the requests that have been sent. So if I go into my FAIR for the demo, that is actually the data which we've just requested today. So I want to take all that data and I want to prove it for use in my workspace. So I'm going to do that, and I'm going to pop it into my data folder. And we're going to confirm. And in the background, that data is actually being moved from a kind of middle storage space into my workspace. So that data now belongs in my project, and I can use it for my own analysis. So if I jump into my files and my data folder, I can actually see the right file, sorry. And this here is a receipt. And that receipt has come with the data. There's a copy of it as well, stored in FAIR. So again, the owner of that data has a list of all the receipts of where all their data has gone, so they know exactly where it is. And as the data receiver, I have a receipt which again includes all that metadata, that really useful metadata that we talked about earlier. So I've got instructions about how I can use the data. I've got descriptions of what's really in the data as well. So to get into it, like you saw, there was a list of all the different data sets, and we can easily transform them into database tables. So each workspace, each project has its own allocation of database space that comes free of charge as part of the project. And if I click into the demographics, so this is the same data set we were looking at FAIR, but we now have access to the line level data. For the purposes of today, we're using a synthesized data set. We also support pseudonymized data sets, again, if you're different levels of access depending on the type of study. So I can see here a lot more detail about each line of my data set. And if I go into analyze data on the right here, I can now see the line level data. Oh, if we ignore that bit because we were doing a practice earlier, there we go. So I can now see the line level data. And maybe I want to get into more depth of information than I could see in that cohort builder that we saw earlier. If I was a braver woman, I would try and show you the same box and whisker chart, but I'm not. So for today, so maybe I want a different analysis. So I'm going to look at the age distribution of this study. And I can do that by selecting age and selecting my frequency bar chart, mostly because I'm not a data scientist and I understand frequency bar charts. So I go in here, it takes a little while to generate, and you can see there's an interesting distribution of the age in my study. And I can also change this. I can, for example, fill that by the sex of the participants in my study. And if I just do a quick rerun, you can see that that will then adapt and change. These are all built using R, which is the data scientist language of choice. The code that is used is then available to you to take on and develop further. This is a very, very basic analysis. But if you wanted to take that and you wanted to run with it and use that, you can do by copying this and then using it further in the platform. The platform itself supports R very natively, but it also supports other programming languages such as SQL. So again, one that we've prepared earlier, if you preferred SQL to R, many people do, you could use the platform to write this. We've preloaded it, but you could be writing this and you could again do much more in-depth analysis of that data set. This is where you can then get, again, you're filtering down this large set of data that you have if you press the button properly. Oh. Let's try that again. I'm using Kara's laptop. That's going to be my excuse.
The second line is the hyperlink.
The what one?
The second line of text. Yeah, if you select it and then you do the run selected, it's the second line of, yeah.
Was that not? Oh.
Product feedback.
Thank you. There we go. So again, here you've got an interesting demographic. You want to dive further into it. Other tools that are included in here are, for example, a load of applications. So things like Jupyter Notebooks, RStudio for the data scientists among you are included as standard. We can provision virtual machines if you want to use GPU on your data, and you can also build your own applications. These give a really nice visual way for your users to get into the data. I'm going to hand back to Kara because I realise I've talked too much, who is going to take you back to the end of the workflow.
Yeah, no problem. I think actually before I'll switch, I'll just do a little plug here for the interactivity of the workspace with something as simple as an R Shiny app, which is kind of like a mini application that's kind of been prepared maybe to show some interactivity inside a data set or be able to show specific results. This links in live with the data inside the workspace. If you update that data, it's going to pull that updated data in, and it's going to let you interact with it in real time. So a little plug for that. But yeah, let's talk a little bit on the machine learning side. So I'm inside a workspace right now that you might see. Maybe it's small. I am in a new hat today. This is the advanced machine learning researcher. So let's assume that's what I am, even though I'm not. And we'll talk about this from the perspective of advanced analytics. So within the workspace, like Laura showed, there's a lot of tools built in, but it's intentionally designed so you can bring tools or bring advanced skills into the workspace. So I've been collaborating with one of my colleagues who is a very talented data scientist, and she's helped to bring in an open source Jupyter Notebook and adapt it for the workspace to pull in data about trying to detect and trying to predict what type of brain tumors are at play. So we've been shared with a Jupyter Notebook here. So this is it in the raw. I've also got some data inside this workspace that is some imaging data itself. So I can view, let's see, some of the apps here inside the workspace. We have an app that's been deployed that is an image viewer app. So I can look at medical images inside the workspace here. Let's take a look at just one or two of these. So we've got some scans here that have been loaded in, and this is also something that can be annotated, and I can make measurements inside the workspace. So especially if I was collaborating with colleagues, this is something that I can save and just share and kind of work with them in real time. But the most interesting part about this, because that's more related to just looking at the images, the most interesting part about working with imaging data is trying to programmatically analyze large batches of data. So if I was to look at this from the perspective of a data scientist who's trying to create some sort of predictive model here, I've already got some information loaded up here. I've got that Jupyter notebook that is focused on machine learning. I won't go through the nitty-gritty on the machine learning model here, but I will go through a couple of examples. So this is a kind of a beefed up virtual machine inside the workspace. So it's kitted out for machine learning. Machine learning can be a bit expensive at times. So if that's kind of one of the projects that's being run, it kind of needs the right hardware for the job. So show you a quick example. This is pulling in that same kind of imaging data. And I can also see, let's look at a distribution of the different types of tumors that are inside the data, both from the perspective of a data set that is for training my machine learning model, and then a data set that is for testing it and seeing how it performed. So I will skip down through a little bit of this to make sure we have some time here. And we'll look at the end here where we've run through, we've created a neural network that has 3 layers to it. So that's kind of like 3 iterations of a model. And what it's done is it's tried to predict the type of tumors that are inside these imaging data sets. So a bunch of numbers here. Unless you're really good at looking at numbers as a human Excel document, it's probably better to visualize this. So if we look at what this looks like, we can see a couple of data points showing up for the first layer, the second layer, and the third layer. And we can also see the training data set, it got progressively better. And the validation data set, the one that I'm testing it on, it kind of did okay. This is one of those cases where you wouldn't want just 3 layers. You'd want 50 or 60 or 100 or way more to get really good at predicting and giving you more true positives. So when I get to the kind of the end here, one of the other things that's kind of interesting to note is just kind of generally looking at how this performs. Like I was saying, if you're looking at kind of the number of true positives versus false positives and so forth, it matters to be able to kind of compare that over that whole data set. And I would look at this clearly and see that there's a couple of these that are not lining up with the right tumor diagnosis and the right prediction. So I'd want to iterate on this and build a lot more into it. So to kind of pause there for a moment, the last thing that I'll mention is just that kind of output of results and being able to cite that back and attribute that to whatever data was involved. Inside the workspaces, you can create an airlock. So that's something where you request to export your results and your findings. And that's something that has to be approved by an administrator. So if you wanted to export your model, export citations, then that's something that can be prepared, requested, and same thing on that kind of data, gateway of access. That's something that can be approved, and you can take that and you can publish that and you can share that with the wider community there. So the last thing that we'll kind of go back to, let's go to the presentation bit, is just a little bit of a plug for what's to come. So I think we have a moment, a moment left.
I'm going to be really brief on this, and instead of plugging what's going to come, I'm going to plug our workshop, which is in tomorrow morning. We have a session where you can come, we can help you sign up, we can help you have an explore around, and we can also show you any features that you would be particularly interested in. The platform itself, like I said, my job is to take feedback from users and to make sure that that makes it back into the platform. We release every 2 weeks, so it's constantly being updated. In the next few months, you'll see, I don't know, some of you might have noticed when Cara was using it at the end there, it looked slightly different, the interface. So that's going to be rolled out a bit more widely. We are currently working on those expanded support for the imaging and the genomics and the other non-linear data types. Looking a bit further into the future, into next year, we will be adding things like advanced version control, as well as the updated data metrics and a dashboard which allows the administrators of the platform to be able to get a bit better view of what exactly is going on. Anyway, like I said, tomorrow morning we do have a workshop, so if there are any questions, I'm happy to take them now, although we have about a minute left. We're happy to take them now, or we are happy to field them tomorrow morning as well. There is links to the platform. They were in the presentation this morning, or just come and grab any of us, and we're happy to show you around. Thank you very much.
Congratulations. I saw this a year, 18 months ago, and you've come a long way. It's great. Well done. The examples that you gave are research-oriented, which is, the explicit stated purpose, which is great. My question is, to what extent could or are you set up to use data that may then be used for regulatory purposes?
That's a good question. I think actually that's one of the ideas of the platform is that there's not just the kind of the use on the analytics side, but there's also use of that collaboration aspect where you can invite people in. So it certainly translates. I think it's kind of a team sport for that. There has to be a little bit of kind of give and take on both sides of willingness to try and willingness to experiment with that, because that's not been the status quo, obviously. But that is one of the things that the platform could lend itself to, because you've got the data and the analysis in the same environment. You don't have to recreate that. You could invite someone in to rerun it and validate that the data is what it says it is, and that it's not been modified, and that those models can be run on it. So I think that's a very potential direction that this goes. But as I said, I think it is a team sport. So yeah.
This is really fascinating. Thank you. So my question is, what are your plans to expand this database to include other diseases? I know you went into a lot of detail, so I'm sort of asking the 30,000-foot question. I specifically am asking because ALS is kind of the new kid in the ALS in the CPath family, and wondering sort of where we go from there.
Yeah, I think one of the plans long term for that, you'll probably hear some other folks today talk about it a bit, but plans long term is to bring more kind of consortia onto the platform, have more data that's available and shared, and to not just limit this to some of the data sets that you saw there, but also talking about some of the other CPath initiatives, and especially data collaborations that have gone on, and try to make something a little bit more kind of expandable and extensible for the communities at play. So I'm not the kind of the CPath person that gets to coordinate some of that, but I would highly recommend asking them, because we've got some people here for that. So thank you very much.
I mean, we are absolutely expanding the platform to include other data. It's going to become a version of this platform is going to become the core data platform for all of CPath data. Some of the older data sets may remain on Coder, but we're transitioning all data over to there. And we're starting to load CP-RND data on there, which is probably going to talk about more.
And just to add to your comment with ALS, we're working with the AMP architecture group. We're trying to figure out who's going to do what. I will say my biased opinion is, they would do omics and we would do where our strengths are in clinical data and link them together in a functional way. That would be most rapid to starting us up. So that's our hope.
And I think the short data, the short answer just to piggyback on both of those, is that this is not only for specific data. This is for all data. That is the goal. As many disease states as we can get in here, long term is what's going to help us really to be able to aggregate that data and use it in the way that we want. So this is by no means at all limited to what's in there now, targets. I mean, really the long-term plan is exactly that.
And just to add to that, I think one of the things that's beautiful about RDCA-DAP is because there's a lot of learnings in looking at rare disease data from lots of different domains. And the next session will talk a little bit about that from a strengths perspective with 2 disease states that we're looking at. But I mean, there's a lot of capacity, a lot of learnings. There's a lot of applicability to regulatory science that's housed within this data set. So our hope is to continue to expand it into the ALS community.
All right. Thank you so much.
Okay, thank you guys so much. So next we want to welcome up to the stage Nicole Vasilevsky, which I can usually say no problem until, of course, I'm on stage. and Ramona Walls, and they're going to talk a little bit about ontology applications in rare epilepsies and Friedreich's ataxia. So welcome.
Okay, given that we're starting late, do we take the normal amount of time? Okay, thanks. Okay, well, thank you all so much for this opportunity to give you some updates on the work that we've been doing with our ontologies and knowledge graph over the last year, as well as where we're going now. I want to briefly acknowledge all of the team who's been working on this over the past few years, including Ian Braun, who you saw in the last presentation, who's done much of the knowledge graph development, but also Daniel Olson, Emily Hartley, and all of our data managers who are working on this project as well. So Nicole and I are going to there we go. Nicole and I are going to tag team on this presentation, so I'm going to hand it off to Nicole for, oh, you have your own mic, great. I'm going to let Nicole start by giving you some background on ontologies and knowledge graphs.
Thanks. All right, so how many people here are familiar with ontologies, since we've been raising hands? Okay, I'll give you.
I'm impressed.
Yeah, that's great. I'll give you a bit of a background. So an ontology is a data standard, so there's been a lot of talk these last couple of days about the needs for data standards, and ontology is a type of data standard that's unique in a way because it's represented in a computational language that allows for computational reasoning and inferencing across data. So an ontology represents information in a specific domain or area of knowledge, and what's unique about an ontology compared to other types of standards or controlled vocabularies is that it defines the relationships between concepts in a domain in this computational manner in a way that we can do computational reasoning across the data.
I think the big one, yes.
So an example of an ontology that we work with at CPath is the human phenotype ontology. So this is an ontology that represents abnormal phenotypes represented in human diseases, including common diseases and rare diseases. So in the human phenotype ontology, there's over 16,000 classes representing the top-level class phenotypic abnormality, and then there's subclasses or children classes of this top-level class that are arranged in this hierarchical classification, which are describing different phenotypic abnormalities. So for example, we may have a class like increased urine alpha-ketoglutarate concentration, which is a child of the abnormal class. So all the characteristics of the parent class are inherited by the child class. So this allows for this kind of subsumption reasoning across the data. We also use external ontologies or other ontology concepts to logically define the terms. So like the case of hyperglycemia, we would computationally define that as a type of increased glucose that occurs in an anatomical entity in the blood. So we're working with a group called the Monarch Initiative that is it's a group that's creating a discovery platform that integrates cross-species disease and phenotype data in a platform. And they've created this gold standard phenotype profile using ontologies. So the gold standard phenotype profile contains over 7,800 rare diseases and phenotypes annotated to these diseases. So these are considered the phenotypes that are typically associated with each of these rare diseases. So these phenotype descriptions are annotated to HPO, or Human Phenotype Ontology terms, and they come from resources including OMIM, the Online Mendelian Inheritance of Man, Orphanet, the Rare Disease Ontology, and Decipher, which includes copy number variant diseases. So we have this corpus of information with phenotypes that are diseases with the phenotypic descriptions associated with them. So we are using this information to build a knowledge graph. So a knowledge graph is a graph database that's a collection of concepts which are represented as nodes in the graph, such as diseases or phenotypes, and edges, which are the relationships between the concepts and the edges, and the nodes and the edges. The edges are the relationships between the concepts. And we can also integrate individual patient data into the knowledge graph, which allows us to query across this data and derive new information and answer questions about this data. So we're hoping that we can add value to individual patients and people living with rare diseases by integrating this patient data into a knowledge graph and enabling querying across this data. So we also are building the CPath ontology, or the critical path ontology, which is an application ontology that's integrated into our knowledge graph. And this is an application ontology that's built for the specific purpose of driving our knowledge graph. And it reuses many terms from existing ontologies to represent types of concepts in this domain. So we have information about diseases, phenotypes, laboratory measurements, and functional tests, so we can standardize the data that we're gathering in the RDCA-DAP and be able to standardize it using this ontology. And this is an open source ontology, and it's available at the URL listed here.
Okay, so now that you're all experts on ontologies and knowledge graphs, or at least have some basic understanding of them, you're probably wondering what we're doing with them in the RDCA-DAP. So that's what this next section is about. The key thing that we're doing is building our own knowledge graph. And what this knowledge graph does that is different than many of the existing ones that are out there already is that it integrates the individual patient level data that we have access to through CPath, through our consortia and RDCA-DAP. That's a really unique aspect of it. We're integrating that data with the existing data from other knowledge graphs like the Monarch knowledge graph that provide a real wealth of information about phenotypes, genes, diseases, a number of sort of known facts and associations, but all at the population or genotype level. So by bringing together this wealth of knowledge that already exists at the population and genotype level with our individual patient level data, we're able to do things like enhancing data integration and discovery across diseases, identifying cohorts or subsets of patients within heterogeneous diseases, translating back and forth across standards for different use cases, supporting diagnoses, and suggesting possible outcome measures for diseases that lack one. This is a screenshot of the portal where you can go to query, where one can go to query the knowledge graph. We have not yet made it public because it does contain patient-level data. We're working on ways to set up access permissions so that users of the RDCA-DAP portal will also be able to go in and do those queries, sort of the same way you do with the cohort builder that Kara showed, where you can do queries and get generalized results without seeing the patient level or line level data at one point. This screenshot, I'm sorry, the text is really quite small here, but this shows the number of annotations that are, or associations that are currently included within the data, within the knowledge graph. We've only got a few thousand patients in there at this point. They're coming from a rare cancer desmoid tumor from the DTRF, rare epilepsies from the Camden-Wren patient organizations, as well as some of the data set from the Friedreich's ataxia integrated database that you saw earlier today. There's also a synthetic data set, the Synthea data set that's widely used that you can use to do some exploratory analysis in the knowledge graph. And so even I can't read those tiny little legend on this figure, I realize now, but this just shows you that there are different associations in the graph between patients and drugs, people and diseases, people and the one all the way on the right, the sort of light green bar, aqua bar, is the associations between patients and phenotypes. And you can see that's the data that we have the most of because those are the associations that we annotated and created through our own data sets. Because the knowledge graph is built on an ontology that has a hierarchical structure, and we're using the same ontology across multiple data sets, we can start to aggregate and do queries across data sets. So what this graph is showing you is that looking at all of the data sets, that is the cancer, the epilepsies, and the Friedreich's ataxia data. Again, apologies for the very small text on the margin here, but these are showing high-level anatomical systems from the anatomy ontology. And if you look at all of our data sets, you can see that most of our patients have phenotypes that are associated with the nervous system. There are also a number of patients that have phenotypes associated with the respiratory system and some within the digestive or gastrointestinal systems as well. Now, if we break this down into individual diseases, we could look, for example, at the tumor patients from the DTRF data set. And you'll see that most of the patients have phenotypes that are only associated with the respiratory system. It's a cancer that occurs, tumors that occur within the lungs. There are also a number of phenotypes associated, again, with the digestive or gastrointestinal system. In contrast, if we look at the Friedreich's ataxia data, these are phenotypes taken from the mFARS rating scale. Most of the patients have phenotypes that are associated with the respiratory system, with the nervous system, not surprising that's given that it's a neurological disease. So having data aggregated at this level, this is a very high level example, but as we get more data and we'll be able to do much more complex queries. And just to point out, this are charts that were generated on the fly given a query, saying, show me this, and this is what it spits out. Thanks to Ian, who can write the wonderful code to do things like that. And so, you can say, look across all of the data within RDCA-DAP, find me all of the patients, regardless of where their data came from, who have an abnormality of their limbs, have a muscular disability, any kind of phenotype that you want to query across at all different levels of organization. And again, because of the hierarchical nature, it will aggregate. So nervous system is including all of those phenotypes that have anything to do with the nervous system. But you can drill down and get more specific if you want to. So before I go into, and Nicole and I go into some of the specific examples of analyses that we're doing with this knowledge graph right now, I want to talk a little bit about a technique that we're using called semantic similarity analysis. Semantic similarity analysis uses the knowledge that's encoded within ontology, that sort of node and edge relationship, and the semantic reasoning or computerized reasoning that you can do with those graph databases to create a matrix of similarities among all of a certain type of entity within your graph. Now, that could be between all of the diseases, it could be among phenotypes, it could be among people. On the example that I've got here on the right, I'm showing an example of semantic similarity analysis on anatomical entities between the renal glomulurus and the glomerular capsule. Now, renal glomulurus is inside a glomerular capsule, and there are many different ways that you can calculate semantic similarity, but essentially they all use this concept of how much of that graph, the paths within that graph, do the two entities share in common. And if you look at, again, small writing, boy, the two different terms, look in the middle, those are all of the terms and the nodes within the graph that they share. And so, not surprising, they share a lot of nodes, they have a lot in common, so they have a very high semantic similarity. If I were to do something like compare renal glomulurus with alveoli, which is kind of a similar structure but in a very different part of the body, there would still be some overlap, but less of it, and so they would have a lower semantic similarity. So what we've done within our knowledge graph is we've taken all of the phenotypic profiles, that is, for every patient, we've recorded all of the phenotypes that we know about them, not just a single one. We've put those into the semantic similarity analysis, and we calculated a large matrix of similarities among all the patients. So how similar is, if Nicole and I were patients, how similar is my phenotype to Nicole's phenotype? Pairwise by every possible set of populations within there. Then with that data, we can do all kinds of different things. And so we're going to give you a few examples of the things that we're doing with them. This is to present a high-level view of one of the things that you can do, which is just visualizing the data. The matrix, of course, because it has many phenotypes, is a multidimensional matrix. But there's a tool that I'll talk about in a few slides that can compress that matrix down into 2 dimensions for visualization, similar to like a PCA, except it's using very different methods, and then cluster those results for visualization. So you can see here based on the parameters and how many clusters, et cetera, this is clustering all of the data. This includes the synthetic Syntheia data set. That's why there's many different points. That's the cloud on the left. So if I click here, it will show you the exact same set of data points, but color-coded by diseases. It's a little hard to tell, but all the way on the right side, there's a slightly bluer green. Those are the Friedreich's ataxia patients. The dark blue near the bottom middle are the desmoid tumor patients. That yellow little oval, not a rectangle, is our rare epilepsies, and then the larger green cloud is all of the Syntheia. This is not showing their clusters, it's just a sort of manual color code, not manual, a scripted color coding of the diseases, but you can actually use this to see that the clusters correspond to the different disease data sets to a large extent within what we've got. Okay, so diving into a little bit more specific detail, I'm going to look at some of the Friedreich's ataxia data and the work that we've used this knowledge graph to do. some of the initial analyses. So the modified Friedreich's ataxia rating scale, I don't think I need to go into a lot of detail here because we had some great presentations on this yesterday that talk about what mFARS is. But one of the key things I want to point out is that as we were mapping the Friedreich's ataxia rating scale to ontologies, first of all, this was a proof of concept to just even show that we could do this. So taking something that's a question and answer pair and putting it into ontology is not a trivial task. So yes, we showed that we could do this and we could come up with things that make sense. We took each question and associated it with the biological trait that it was trying to get at. Was it motor function? Was it speed, et cetera? And also the anatomical entity that it was associated with, that a limb, the face, the gastrointestinal tract, et cetera. So we've got that data. The steps that we took to do this analysis were that we first evaluated the whole COA instrument. We modeled the instrument components and questions to ontologies. We used many of the terms that we needed to describe them were not in existing ontologies, so we worked with those ontology developers to create new terms where they were needed. We ingested all of the data and all of the ontologies into our knowledge graph, and then we did queries across that knowledge graph. And one of the pieces that we got out of it was a semantic similarity matrix that I showed you a minute ago. And here's that same matrix, focusing just in on the clusters that for FA. These are the same, they look a little bit different in this graph because they've been pulled up, but it is the same exact clustering approach that was used for all the data. We didn't recluster just within the FA. It's from all of the clusters. You can see, as you might have noticed in the other graph, there were two clusters within Friedreich's ataxia. This is the method, the T-sign distributed stochastic neighbor embedding is the method that does the clustering, and then the dimensionality reduction. And Ian can yell at me if am I getting that right? Does T-sign also do the dimensionality reduction? Thank you, K means clusters, and the T-sign just does the reduction of dimensions then. First I saw this as, wow, this is really exciting. We've got two really distinct clusters of FA patients. What's going on here? Well, it turns out that, cluster 1, the blue cluster, is an individual data set. This does not include the FA comms data set within here. So it includes the other 5 data sets that we have available for Friedreich's ataxia right now. And it only includes the patients that had data on the particular, on mFARS within there. And it would be cool. So it turns out that cluster 0, you can also get, it will also report on you which ontology terms are enriched within each of those clusters. And it's kind of cool because cluster 1 has a lot more enrichment for facial abnormality phenotypes, whereas cluster 2 has a few facial phenotypes enriched, but more along upper, like limb abnormalities. But we don't actually think that's a big biological difference. We think it's just what one data set collected versus what the other data set collected. And to me, this is so, like, it's confirmatory. This is what we wanted. We didn't expect really earth-shattering results with this initial analysis because FA was fairly well known. We actually wanted to use a data set where we could have some predictions about what was going to happen and see if they were confirming those. So they just asked different questions in one survey. But the nice thing about this is when you're trying to look at a data set for initial analysis, it gives you another way to visualize it and say, where might I have problems, right? Do I want to include this data set number 6? Because maybe it's different than all the other ones, and this is a great way to look at that. All right, I'm going to hand it off now to Nicole to talk about some of the work we've been doing with rare epilepsies.
All right, so we have some data coming from 2 data sets from rare epilepsy groups. And we did an analysis to try to look at the semantic similarity across phenotypes for these patients. And we wanted to kind of do this proof of concept where we had done some research like this initially. This is work that was done by the Monarch Initiative. So I'm just going to give you an example where we know that we can actually use the semantic similarity metric to diagnose patients with rare diseases, and then show you some examples of work we've done at CPATH to try to utilize some of the individual patient data that we have to compare phenotypes from the gold standard phenotypes to our actual individual patient data. So this example here, like I said, is work done by the Monarch Initiative, where 2 patients came to the clinic and they presented with mutations in the same gene, but they had varying phenotypes, and these patients were undiagnosed at the time that they were seen in the clinic. And these patients had a collection of phenotypes that are described on each side in green and in orange, and these patients didn't match up any typically known disease. But looking at the gold standard phenotypes for this Wiedemann-Steiner syndrome in the middle in blue, these are the typical phenotypes associated with this disease. And these patients didn't match these phenotypes exactly, but some of them had some fuzzy matches or some similar overlapping phenotypes with the gold standard. Like, for example, the patient on the right, the boy, had a long toe, where the typical phenotypic presentation is a short toe, but these are both abnormalities of the toe. So if you were looking at the hierarchy, you could see that these are in the same branch of the ontology. So using this phenotypic similarity metric, the physician was able to diagnose these patients with actually having the same disease, even though they didn't have the typical phenotypic profile. So we have this individual patient data at CPath coming from 2 individual data sets, one the CAND data set, which is the KIF1A associated neurological disease, and a set of patients from patient registry from the Rare Epilepsy Network. So these surveys describe patient diagnoses and their accompanying phenotypic profiles. So we leverage the Monarch Initiative Knowledge Graph and gold standard disease to phenotype annotations and performed semantic similarity analysis against our individual patient data. So our individual patient data was mapped to these ontology terms. We annotated to the ontology terms. This is work done by Daniel Olson in our group. And then Emily Hartley, one of our data engineers, performed this analysis to first compare if the patients in the CAND data set, the CAND disease is a group of disorders which include some common diseases like autism and epilepsy, and also some monogenic diseases, which are described here on the bottom of this graph. So we looked at the phenotypic presentations in the patients from our data sets and compared them to the phenotypic presentations that are typically associated with the patients from the gold standard data set that we had. So the numbers on the bottom correspond to the percent of similarity between the patient data and the gold standard data. So if we look at the center example, the intellectual disability autosomal dominant 9, we can see that there's dozens of patients that have between 50 to 60 and 60 to 70% similarity to the phenotypes that are typically associated with this disease. And if we look at the other 2 diseases, we see that these patients don't have a lot of phenotypic overlap with these diseases, suggesting that these are probably not the diagnoses that these patients have. Also, looking at the REN data set that we had patients who had reported that they had known diagnoses or had been diagnosed with a certain disease, which are listed on the x-axis of this graph. And we compared the phenotypic profiles from the gold standard that are typically associated with these diseases, listed on the x-axis, and compared the number of patients who matched the phenotypic profiles in the individual patient data sets to the gold standard. So like looking at a Cardi syndrome all the way on the left or going towards the center, the LGS, the Lennox-Gastaut syndrome, or West syndrome on the right, we can see that there's, again, dozens of patients who have over 60% or up to maybe 90% overlap with the phenotypes that are seen in these profiles, whereas a lot of the other patients, and there's also a lot of patients in these particular diseases who have differing phenotypes from what we see in the gold standard. We're also able to view this data using a heat map as well, using this method called PhenoGrid. So here we have an example where on the x-axis on the top, this corresponds to each individual patient, and this is for one particular disease, tuberous sclerosis complex. And so on the y-axis on the left, these are all the phenotypes that are associated with this disease. And then the darker colors correspond to high semantic similarity between the 2 phenotypes in the patient data compared to the gold standard. And the lighter colors, the green boxes, indicate lower semantic similarity. So if we look at the first patient, we can see that they have a lot of similar overlapping phenotypes to the typical phenotype seen in this profile. Some of the blank spaces indicate they don't have that phenotype at all. So, and then if you look at the darker colors, like on the right, the infantile spasms, This has a 98% similarity where the patient reported they had myoclonic seizure, but the gold standard says they have infantile spasms. That's because these are basically siblings in the same branch of the same hierarchy. Whereas at the bottom, the shagreen patch, the gold standard, this is the gold standard phenotype that's seen in these patients. And one of the patients reported they had dental enamel pits, but this is only 36% similar because they're both, if you go up the hierarchy, they're both types of abnormality of the connective tissue, but they're in different branches of that hierarchy, so they're actually not very similar, but they do have kind of a high up common ancestor. So it was really interesting to see these results, and we definitely have more querying and analysis we want to do with this data, but our initial takeaways from this are, one, there could be gaps in knowledge, and this gold standard data set is what we're working with, but there could be really other gold standard phenotypes that are associated with these diseases. So we would like to look into that further. We also took this from patient registry data, and it's really hard to determine if the survey questions in the registry data cover all the phenotypes that are in the gold standard. So that is something that we also plan to follow up on and look and see if the registry is covering all the phenotypes that we are intending to examine. And another thing about the registry data is that it's not also meant to capture the natural history of the disease. It's meant to capture the disease status at a certain point in time. And it's also kind of on a rating scale. So it's not always capturing the exact phenotypes that are representing the disease in itself. It's more representing the patient's reporting at that time point. And there's also a lot of phenotypic noise. So patients may have a rare disease and present with certain phenotypes, other co-occurring disease that may present with certain phenotypes. And that can be reported on the registry data, which may be irrelevant to the actual rare disease that they have. And then also there's been difficulty with the mapping, doing the mappings themselves. The survey questions can be very vague and sometimes hard to interpret, so there's a lot of subjectivity in doing the mappings themselves.
Thanks. All right, so just to wrap up where we're going to go with this and you know, what we expect to do in over the next year or so, we're going to addressing the semantic similarity issues that Nicole just outlined in the previous slide. We want to do different activities to identify and fill the gaps in the rare disease phenotypes within the natural history. That could include analyzing real-world data and then contributing that knowledge back to open open phenotype descriptions like the Monarch graph. We want to work with communities to standardize data. I'm really excited yesterday to hear several people recommending that we start to standardize surveys and COA questions. We'd love to give input on how you can use ontologies to do that. We definitely want to reduce the noise that we're getting. One way to do that would be to provide a good mechanism for different individuals, whether they be patient groups or industry or even individual practitioners, to contribute to gold standard phenotype profiles when they know them. We want to improve our mapping efficiencies. There's a lot of work to do here. You know, in terms of the COA analysis, like I talked about, that the FA data was a proof of concept for. There's a number of ways that we would like to build off of that. We are currently mapping 4 different instruments to the CP-RND ontology, and of course we're also reviewing all of those wonderful resources that Lindsay talked about in the RDCA-COA resource yesterday, the COAs that are there. We want to do additional semantic similarity analyses that include phenotypic descriptions from existing COAs, but also possibly include additional types of phenotypes from labs or imaging. And we would love to incorporate longitudinality. The FA data that I showed just treated each time point as an individual point, that we get the same results when we cluster by patients. Long term, we would love to have a sort of a COA recommender. We've had a number of conversations with Lindsay about this and how we can make this happen. You know, that would help you, that would build on top of the resource that's already there from the RDCA-COAC to that's a real mouthful, isn't it? But help you select based on the phenotypic profile of the disease that you're interested in, would help you identify other COAs that might be useful. We think that there's a lot of potential utility. We've been doing this now for about 2 years. and it's been kind of slow going because there's a lot of just like we're doing things that are really new. There's a lot of work in ontologies out there, but nobody's been doing it quite the way that we've been doing it. So we spent a lot of time just figuring out how to do this, and I feel like we're really starting to get up to speed. And I hope that the work we've been doing can be of great use to the rare disease community going forward. We'd love to get your input and potential ideas about how you might want to use this within the groups that you're working with. Some of the key uses for ontologies include data harmonization, integration, and discovery. I showed a very high-level example of how you can use ontologies to search across diseases and data sets that would normally be isolated or siloed. Nicole gave a great example of how ontologies and knowledge graphs can support diagnoses. You can imagine using that kind of similarity and fuzzy matching to go into EHR records and search for patients who might have a disease that have not been diagnosed yet. The example that I gave, even though there were not, as expected, there were not really great interesting cohorts or subsets of patients within FA, we are very confident that we'll be able to use this to identify subsets of patients within rare diseases. And then it can even do things like using, when you're looking at inclusion-exclusion criteria, rather than using a single criterion, you can use the semantic similarity or the complexity of the knowledge graph to look at patients as a whole set of their phenotypes, not just as which disease they have. And finally, the COA recommender that I mentioned before is something that we would love to see. That's something that we're going to be spending a lot of time in the next year. And I think we're about out of time. But if there's time, we can take questions, or if not, if we're at the end of the session, I'm happy to stick around during the break for any questions that people have.
We can take a few questions.
Okay, so we've got time for a few questions.
Hi, thanks for that interesting presentation. Doug Langman, University of Iowa. I was wondering, with your gold standard phenotypes and semantic measurements of semantic similarity, is there any weighting involved in terms of the importance of various features of a phenotype? I mean, many diseases have certain features that are necessary or possibly sufficient, or we can talk about that in terms of sensitivity and specificity of different features of a disease. Is there any attempt to...
I think, Nicole, you can answer that best.
Yeah, so the gold standard phenotype annotations from the Monarch Initiative actually include frequency information, like how frequent the phenotypes occur in the patient data or in the patient populations. However, in the analysis that we did, there was no weighting for the frequency. So that is something else that we would like to take into consideration in our future analyses.
Okay, thank you.
My question, and I don't know if this de-identifies, I loved the semantic, the diagnosis, I loved all that. And one thing that I was thinking is behavior may play a role, because how I acted when I was growing up in Texas is not how I acted in Boston, and it's not just because I'm older. So can you break this down and say, let's take a cluster of people in certain geographies and see if this is similar, because I wonder if my behaviors, hanging out outside versus staying indoors, coats and stuff like that, is also going to modify the outcomes that you're seeing.
That's a great point. Yeah, that's something that we haven't considered really, but it would be lovely to include that into the analysis. And with the knowledge graph, we have the ability to incorporate other kinds of modalities too, so that's something we should definitely consider.
So there's a couple of challenges there. I mean, one is, do we have the data to do that? The second is, do we have the ontologies? And so I've done a little bit of work on ontologies for social determinants of health. The environmental health community is particularly interested in that. But I would say that Nicole and I have both been very active over the years in the Open Biological and Biomedicals Ontology Foundry, or OBO Foundry, where many of the well-known ontologies like the Human Phenotype Ontology, the Gene Ontology, the Uberon Anatomy Ontology sit. Though there are not, those are a great set of ontologies to work with, there's not a lot on social determinants of health, on behavior, on environment. There's a wonderful environment ontology, but it doesn't describe the kinds of environments that you're talking about. So before we can make progress, like what we're doing now is taking advantage of the ontologies and the kind of knowledge that already exists. It will be another 5 years before we can start to add in those other data sets, unless somebody wants to give us $1 billion, then we can build the ontologies.
The knowledge graph and ontologies are open too, so you're welcome to do the studies yourself too.
I have a quick question, almost to dovetail from the conversation that was just started earlier. It's like, if you could apply this to ALS, your ontology, we have all this omics data, we have, income data, we have natural history studies. How would this, what would you be able to do with the ontologies work that you're doing?
Well, I think that one of the ALS would be genomic diseases are great, especially when when the variants are known, because there is so much information on how different genotypes are related to proteins, related to pathways, related to expression. There's a lot of information out there, so you can kind of plug your gene variants into the existing Monarch graph, get the information that Nicole, you can like slap me if I'm misrepresenting, but this might, you can get you can use the Monarch and other sources to have access to that wealth of omics data that's out there, and combine that with individual patient data. That's where we would love to go with this eventually. Thanks. Oh yeah.
Yeah, hi. I just, Jackie French from NYU. I just wanted to add to the gentleman from Iowa in regards to weighting, and that is that, for example, in the rare epilepsies, almost every rare epilepsy has like 5 things in common. They have developmental delay, they have autism, they have, multiple seizure types. I could go on and on. Sleep problems, behavior problems. So it's really, when you look at those graphs, you're going to see lots of things that are the same, but can you sort of sort out the things that are actually different so that you don't just say, oh, all these things are the same?
So I think that really addresses the fundamental issue that Nicole, as we were preparing this talk, something we were really aware of was that the way the data are collected, the purpose for which they're collected is one thing, right? So if it was, for example, the mFARS rating scale, that was designed and validated specifically to understand where someone is in a progression of Friedreich's ataxia. It's not designed to describe the full phenotype of every person who has FA. Likewise with epilepsies, right? The registries are collecting data for a particular purpose to understand what may be important. They're not collecting data for the gold standard of describing that phenotype. But I've heard over and over again over the past 2 days how important it is to understand the natural history of all these rare diseases. So maybe we need to look to the people who are collecting data and say, can you start gathering, or can we minimize the burden on patient and start getting that information from real world data? Are there other ways that we can start to really understand the natural history of all of these rare diseases through the ontologies?
And combining it with genetic information too would also be really valuable if we had information about the genotypes of genes affected in different variations. That would also be relevant as well.
Yeah.
Just a comment for everybody. I'm reasonably familiar with the ontology field. I can tell you Ramona is the single only person in the entire world who can excite an audience around ontology.
Sorry, Nicole. Nicole's pretty good at it too. I've seen her give talks.
On that note, warm thank you to them for their presentation. Thank you. Okay, so at this time we're going to go ahead and break essentially for a transition. We have about just over 15 minutes until the breakouts start. So just a quick reminder, and those start at 3:45. So we have the digital health technology case study in rare disease trials upstairs in the junior ballroom in Salon 1, so the first room. We have the increasing diversity and access for clinical trials. That's going to be over behind the elevators here on this floor in the London room. Then we have novel approaches to trial designs for rare diseases, which is right next door here in Ballroom E. And then rare infections, new endpoints to inform trial design is also upstairs on the second floor junior ballroom in Salon 2-3. So we'll have those breakouts. Go ahead and start making your way there. Those will start at 3:45. And then we will come back here for a few closing remarks and also give you a preview of the exciting things that we have for tomorrow.
Yeah, I'm live. Yeah, I'm going to start the closing remarks. Thanks everyone for sticking around. I think, in rare disease, people working in rare disease are warriors, but, coming back for closing remarks after 2 days, you guys are really true warriors. I'm guessing you'll want it to, last for more than 15 minutes, but unfortunately, I'm going to make it a little shorter than that. I think we had a great sessions today of meetings, a little more focus on RDCA-DAP and our platform. There is another day tomorrow, and I know, maybe some of you are leaving, but some of you plan to attend, but you might be thinking, oh, maybe I'll take an earlier flight. Maybe it's going to be sunny tomorrow. I'll take a walk in Georgetown, get some cupcakes or go to Domestic Wine. It's a great wine shop in DC. But we really have, a morning packed with really great sessions. I was in the preparation in some of them, and it's really great content. We will have a session on smarter clinical development using disease simulation with Quinton Health, a French company, and our colleague from CPAF, Cécile Olivier. We will have a consent on data sharing, a session on consent and data sharing with Advara. We will have an interactive session on RDCA-DAP if you want to dig further into what you saw today, presented by the Aurelia team and the CPAF team. We will have a great session with NLORM, Luc Rosen from the KIF1A Foundation, and our own Tim Franson of the regulatory consideration for N-of-1 trial. It's really a very fascinating conversation. And then another session with a session on real-world evidence, control arms in transplant trials with my colleague Amanda from TTC. A session on consideration of global regulatory considerations with Michelle Campbell and Hong from our team. A session on RDCA-DAP with Lisa from Nord, taking a case example from a patient registry that Nord helped establish for Prader-Willi syndrome with some early analytical and a showcase on how we can leverage registry data through RDCA-DAP to help inform registry activities. And then a patient perspective on what it is to collaborate with CPAF. So really a whole lot of interesting sessions again. So please stick around and come back tomorrow morning to finish the conference with us. We had a question today during one of the sessions about what do you do to continue to increase the database. And so data sharing, we just beat the drum on that. We always have sessions, so we didn't feel the need to have a particular session on that today. But it is our day-to-day. It is weekly effort that I'm committing and the team is committing to discussing and trying to increase our database. There is new avenues that we're trying to to increase the amount of data, rare disease data shared to RDCA-DAP. That includes federation of the platform with other partners. We may finally have a breakthrough, but I'm going to knock on wood and not say anything more. I'm really hoping it's going to pan out with a federation with a platform that has a lot of industry clinical trial data. Also, something else that we're trying is we are under negotiation right now with a rare disease company that shared data before to us, and we're moving to try a master data contribution agreement. So not a single data set, but really trying to build an agreement to share more data right now and also committing to share data in the future and facilitating that data sharing through that master agreement. So again, another knock on wood that it's going to pan out. I think it's the kind of commitment that we need for an effort like RDCA-DAP to really work. We really need, to think prospectively on continuing to share data to RDCA-DAP and work to build consent to facilitate data sharing. And again, we'll talk about that tomorrow. So lots of effort, and we're continuing to work every day to increase the database and be able to leverage that database to help facilitate rare disease drug development. It's one thing that I heard recently, someone said, CPAF, they want our data, and it's not exactly that. We don't want your data. We need your data because we need data to be able to leverage it to develop the solutions that we work on at CPAF. So on that note, thank you again for sticking around today, and I hope to see you again tomorrow. And have a great evening.